Hello students, in today's class we are going to learn about isolation and enumeration of microorganisms by pure culture methods. And here we are going to discuss about two pure culture methods, Foure plate and spread plate method. So the aim of the experiment is to isolate and enumerate microorganisms from the given sample by pure culture method. Requirement for spread plate method includes LV agar containing Petri plates, glass spreader, alcohol, sterile saline in tubes, and the sample. For Foure plate method, we need sterile empty Petri plates, sterile 30 ml molten LV agar in tubes, glass spreader, alcohol, sterile saline in tubes, and the sample. Coming to the principle for both methods, so in spread plate technique involves using a sterilized glass spreader to apply and spread a serially diluted sample of bacterial suspension over a sterile nutrient agar plate. The plate needs to be dry and at room temperature so that the agar can readily absorb the moisture. Later, after incubation, each bacterial cell will develop into one colony. A successful spread plate will have countable number of isolated bacterial colonies that are evenly distributed on the plate. Next is Foure plate method, which is based on the fact that when agar medium is mixed with microorganisms and is incubated, each viable microorganism will multiply to form a separate visible colony. In this method, certain volume of a serially diluted liquid sample is mixed with about 25 to 30 ml of specific molten agar medium, which is at about 40 to 45 degrees Celsius, and then they are transferred into a Petri plate. Following solidification and incubation, the viable microorganisms in the sample will grow such that each viable cell will form a single colony, and the number of colonies obtained can be calculated as colony forming units per ml. So coming to the protocol for serial dilution of the sample, you need different tubes that will be labeled either 1 to 10, 1 to 100, 1 to 10 to the power 3, and so on dilutions, or you can also represent the first tube as 10 to the power minus 1, 10 to the power minus 2 for the second tube, and so on for the tubes. Then you need to transfer 1 ml of sample to the first tube, and here your dilution is going to be 1 to 10 dilution, or the dilution factor will be 10 to the power minus 1. After mixing the sample and the diluent or the saline, we need to transfer 1 ml of sample, diluted sample from the first tube to the second tube. This will lead to 1 to 100 dilution in the second tube. In this way, we mix and transfer 1 ml of sample to the subsequent tubes to complete the serial dilution method. So in case we are making use of a solid sample, then we need to measure 1 gram of the sample instead of 1 ml. Next is the protocol for the spread plate method. So for spread plate method, we are going to select any of the suitable serially diluted sample, take 100 microliter of the serially diluted sample and transfer it to a sterile Petri plate. Then we are going to sterilize the glass spreader by dipping it in alcohol and flaming. After cooling down the glass spreader, the culture is spread uniformly on the surface of the Petri plate with the help of the sterile glass spreader. Following this, the plates are incubated at 37 degrees Celsius for 24 to 48 hours, and the number of colonies obtained on the Petri plate are counted and the results are interpreted. For Foure plate method, we are going to similarly choose a suitable serially diluted sample for further procedure. We need to take 100 microliter of the serially diluted sample and transfer it to a sterile molten LV agar in tube. The temperature of the media should be around 40 to 45 degrees Celsius. So this temperature will allow the media to be molten, and at the same time it will be bearable for the microorganisms to survive. Immediately, we need to mix the culture with the media and transfer the contents onto a sterile Petri plate. Followed by this, the Petri plate is incubated at 37 degrees Celsius for 24 to 48 hours. And then again, the number of colonies obtained are counted on a Petri plate and results are interpreted. So coming to the result interpretation, so here we first need to make a observation table that is going to contain your first column is going to be your tube number. The second column is going to be the volume of sample. Uh, so here you are going to represent how you're going to do the mixing. So the second column is volume of sample and the third is volume of saline. So for example, in the first tube, you are taking 1 ml of a sample from the flask and you are taking 9 ml of the sterile saline. So here, since you are mixing 1 ml with 9 ml, after mixing the total volume is going to be 10 ml. So the dilution factor becomes 10 to the power minus 1, or you can say the dilution is 1 to 10 times. So similarly, in the second tube, you are going to mix this and transfer 1 ml to the second tube that is already containing 9 ml of saline. So your dilution is going to be 1 to 100 for the second tube, and the total dilution factor is going to be 10 to the power minus 2. Now suppose if after plating, suppose in your experiment you have used dilution factor tubes 10 to the power minus 2, minus 3 and minus 4 for plating, and the number of colonies that you have obtained in the 10 to the power minus 2 tube. When we have used the 10 to the power minus 2 dilution and you have plated 100 microliter of the diluted sample from this dilution onto a Petri plate by either spread plate or Foure plate method. So if suppose we have got 900 colonies, then how many colonies will be present in your 100 microliter of undiluted sample? To calculate that, you need to multiply 900, that is the number of colonies you obtain, multiplied by dilution factor. So here, since the dilution is 1 to 100, so you need to multiply it by 10 to the power 2. So the negative symbol is going to change to 10 to the power 2 here. So there will be 900 into 10 to the power 2 cells in 100 microliter of undiluted sample. However, we need to calculate how many colonies or cells are present in per ml of the sample. So here we further need to multiply by 10. So that will, since here you are having number of cells per 100 microliter and we need to find out a number of cells in 1 ml. So you further need to multiply your answer by 10. So that will give you 900 into 10 to the power 3 cells per ml. Now, similarly, suppose in my third tube that is having a dilution factor of 10 to the power minus 3, I have plated 100 microliters on my Petri plate, and after incubation, I got 94 colonies. So that means in my undiluted 100 microliter of sample, there are going to be 94 multiplied by the positive dilution factor, that is 94 into 10 to the power 3 cells. So these are going to be present in 100 microliter of the sample. So if I want to find out how many will be present in 1 ml of the sample, then again I further need to multiply by 10. So the value is going to be 94 into 10 to the power 4 cells. Similarly, in the fourth tube, which is having a dilution factor of 10 to the power minus 4, if I am getting 8 colonies, after plating 100 microliter of the sample, so that means in my undiluted 100 microliter of the sample, there are going to be 8 multiplied by 10 to the power 4 number of cells. Similarly, if I want to find out how many are present in 1 ml of the sample, undiluted sample, then it is going to be further. I need to multiply the answer by 10. So that will be 8 into 10 to the power 5 cells. Now, once we have these values from different dilution factor tubes, we need to find out the average number of cells that are present. So in this example, you can see that the average is going to be the average of the three numbers, that is here at 900 into 10 to the power 3. I'm changing the factor of all to 10 to the power 4 so that I can easily do the calculation. So the average is going to be 90 plus 94 plus 80 divided by 3 multiplied by 10 to the power 4, which is equal to 88 into 10 to the power 4. So in this way, we can find out or interpret our results to determine how many microorganisms are present in the undiluted liquid sample, or in case you are using a solid sample, then the answer is going to be these many number of cells per gram of the sample. So we can also calculate the colony forming units or the number of bacteria per ml using the formula, that is total number of colonies obtained multiplied by dilution factor divided by volume of the specimen that is used. So that's it about the experiment. Thank you very much, and kindly like, share and subscribe if you like the content. Thank you.